Hi, this is a Science Hangout from Science Mission. I'm Sadashiva Pai. So today we have Dr. Nikos Tapinos with us. He's the Associate Professor in Brown University. He has published several articles in very respected journals. He's in the editorial board of several journals also, and he has received several grants, and he's one of the emerging scientists in the field. So without much further ado, let's welcome Nikos Tapinos. Hi, Dr. Tapinos.
Hi. Thank you for having me today.
Welcome to Science Hangout. Nice having you over. So let's get going. Can you please talk about yourself, tell about yourself, like how did it all start from the beginning?
Yes. So I'm originally from Greece. So I studied medicine in Greece. So in Greece, we don't have a combined MD PhD program. So when I finished my med studies, I always wanted to do research. So in order to do that, I started a PhD after finishing my MD. And I really loved it. And I decided to follow this career, the basic science career. So even though I have an MD, I never, practiced medicine, so I don't see patients. So my PhD, I did my PhD in Greece as well, and it was a little bit different from what I'm doing now. It was molecular immunology. But then after I finished my PhD, I always was fascinated by the nervous system, and I wanted to study the nervous system. So after finishing my PhD, I joined Rockefeller University for a postdoc, and later on the last year as a junior faculty there. And after Rockefeller, I left and started my own lab at the Geisinger Clinic in Pennsylvania. And a year ago, I joined Brown University as an associate professor here.
Okay. So you have a pretty interesting career. And tell us about some of the people who encountered, who helped you, who mentored you, who served you as a role models.
Yes. So one person that influenced but helped me when I started my PhD was my PhD supervisor. So her name is Maria Polychronis. She was just coming back to Greece from Rockefeller University.
Okay.
And she helped me because, from medical school, I didn't have any experience with basic science. So she helped me a lot. So she taught me a lot of the techniques that I used, and also introduced me to Rockefeller, saying that you have to check this place. And after when I went to Rockefeller, even though I cannot really say, I have one person as a mentor. What actually, dictated my career so far is the atmosphere and the intellect at Rockefeller, which is an incredible place. And having going to the faculty club and having a beer or getting a breakfast or a lunch at the cafeteria, and all around you is like all kinds of Nobel laureates and Lasker Award winners and National Academy of Science people. I mean, you look up to it and says, okay, I want to be one of these people. I want my science to be this good. And also Rockefeller, they have, as an institution, I love this, what they do is they don't want you to enclose yourself. So they tell you, okay, think outside the box, push the boundaries of research. And this is beautiful. I mean, this is, I think, what you should do when you do research, and also you should have fun. If you don't have fun, then it doesn't really make sense.
Okay, that's good. And I know you are busy with your research, doing a lot of work, publishing and all. In addition to research, what are the other extracurricular things you do? Like, what are your hobbies and all?
And extracurricular, my family, I have 2 kids, and my wife, we spend time together. We like to travel. And personally, I like to play basketball with some friends. I like to sail. I love sailing. This is basically what I do.
Okay. And coming back to the lab, like just to tell us about your lab people, grad student, postdocs,
Yes.
and
So my lab now, as I said, we joined a year ago here at Brown. So now I have two postdocs in the lab, one technician and two graduate students. So that's basically my lab right now.
Okay. And how do you motivate these kids around?
I think that, again, my field is like this. So I don't think that, actually not I don't think, I don't believe that you need to motivate people to do science. I think people should be motivated to be scientists. Science should motivate them. And the projects and what they do every day should be the highest motivation. So what I mean by that is that they have to wake up every day and be very happy to come to the lab and keep on pushing and keep on doing science. If they don't, then probably it's not the right thing to do. That's how I see things. And in the lab we are very friendly. We have a very nice relationship, all of us together. So the lab it works like my office is always open. So they come in and out all the time, every day. So we go for lunch, dinner together. So we have a very nice relationship with the people in the lab.
Okay. So let's talk about some of the projects you are doing and your work.
Yes. So the work I started again when I started from Rockefeller. I will give you just a small background of my work. So I worked at peripheral neuropathies, and we used a very interesting model at Rockefeller. So we used live leprosy bacteria to infect the peripheral nerve. And because the leprosy bacteria is the only bacterium that actually can infect Schwann cells and can live inside the Schwann cells. And it doesn't kill the cell, it just takes over both the transcriptional machinery, also signaling of the Schwann cell in order to make a very nice home for the bacterium. But this process changes the Schwann cell, what this means, the Schwann cell demyelinates and the Schwann cell expresses different proteins than the normal Schwann cell. So we thought that we can use this as a model to understand signaling pathways that regulate myelination. So after I started my own lab, I'm not working with bacteria anymore. So we continue to work on different signaling pathways that I started identifying at Rockefeller. And at some point, especially one particular pathway which we found it was very interesting that regulates migration of Schwann cells. So we had the idea now 6 years ago to see if this same pathway regulates migration of glioblastoma cells, because we know that in the brain, in an adult brain, these are the cells that actually migrate extensively. So, and this is where my medical background helped. So I reached out to neurosurgery department at Geisinger Clinic, and I managed to get through our collaboration, I got an appointment as a director of the neurosurgery research there. So we established a program for glioblastoma research, which is based on primary cells. So we made a repository of glioma stem cells and differentiated cells from patients, and we started asking very basic questions to understand migration, invasion of these cells, also understand transcriptional responses. So what I'm going to show today, the epigenetic regulation of peripheral nerve injury, and especially RNA epigenetics, which I'm really interested. So we translated this to the brain cancer as well. So we're having a big project with RNA epigenetics of human cancer stem cells. And as we came to Brown, I mean, we keep having both arms of the projects, the peripheral nerve, and especially nerve regeneration and the epigenetic stuff, and the glioma stem cell and glioblastoma research. So this is what we basically do in a nutshell. Yeah.
Okay, can you please go ahead and present your work?
Yes, of course. Okay, So the work that I will present today is actually the role of an Egr2 long antisense RNA nerve injury response. And in order to give you a background of what is and how we started working on long non-coding RNAs in nerve injury. So what is a nerve injury response to the peripheral nerve? So we know that the differentiation of Schwann cells during development is regulated by expression of specific transcription factors. So basically, immature Schwann cells during development receive axonal signals. One of the major signals is neuregulin-1, and this upregulates various transcription factors. Some of them is like NF-kappa beta, OCT6, BRN2, and others, and which sends the immature Schwann cells to the promyelinating stage. And to change then from promyelinating to myelinating, the most important transcription factor that regulates this transition is Egr2, or CROX20. Now, upon injury, there is a rapid upregulation of c-Jun and SOX2 transcription factors and downregulation of Egr2. And in a nice schematic that was published from Christian Jensen's lab, I mean, you see that the immature Schwann cells, these are the transcription factors that regulate the transition to promyelinating cell. And from there on, CROX20, Egr2, and c-Jun are the main players that regulate myelination or nerve injury response in Schwann cells. So we know now from studies of many groups in the field that during nerve injury and after an injury, there are several things that are happening in the nerve and in Schwann cell. So if you want like an overview of these things is that you need to suppress myelin differentiation, because what happens immediately after injury, there is a program that starts which makes Schwann cells to de-differentiate it to an immature state, which is and form this cell type which is called a repair Schwann cell, which will drive nerve regeneration. So the first thing you have is a downregulation of myelin genes and upregulation of markers of mature Schwann cells. Then you activate trophic factors, surface proteins, which provide support for injured neurons. And the Schwann cells, the regenerating Schwann cells, form these regeneration tracts, which is called Wagner bands, and where within these bands, the newly formed, the new axon is going to go and start regrowing, okay? Mm-hmm. So during this injury, again, work for many years and from many labs showed that there are several steps to the injury response. So we have the trophic support decrease during the axonal injury, retrograde axonal discharge, and local injury signals that are generated in the axon. In the Schwann cells, pericardial, we have activation of several signaling pathways. People have shown ERK1/2 and P38, PI3 kinase, mTOR pathway. And these signaling pathways lead to activation or inhibition of transcription factor program, as I said before, c-Jun, Egr2, CBP, STAT3, and eventually other adhesion guidance molecules, neuropeptides, cytoskeletal adapters are regulated to form this, to actually regulate this whole process of regeneration. Now, in this whole process, the most important step is the step which controls the transition between the myelinating Schwann cells to denervating Schwann cells, or repair Schwann cells. And we know that this transition is controlled by a balance of positive regulators, which is Egr2, OCT6, SOX10, BRINC2, and NF-kappa beta, which favor the myelinating state, and negative regulators of myelination like c-Jun, NOT, SOX2, PAX3, and ID2, which actually favor the repair cell and the denervated state. So again, the process is not happening obviously in a day, but there is a time course where this process from injury to regeneration happens. And in the mouse, on the rodent model, we know that what happens in the first days, you have the breakdown of myelin, which followed by Schwann cell proliferation. Roughly in the first week, this is happening. You have axonal degeneration from day 1 to day 3 or 5. Functional recovery happens in 4 weeks, like a month later. And one very early event in the first day is the induction of c-Jun. So we now, where we come is that we were interested from the beginning in this window, which is from the injury to day one. So these 24 hours we think are very crucial. And why we think that? Because these 24 hours will dictate the quality and of the signals and the transcriptional regulation that will activate or downregulated genes, which are very important for continuation of this program. And we know that in these 24 hours, you have the Egr2 downregulation and the c-Jun SOX2 upregulation. But how does this happen? Yes, we have signaling, we have intracellular signaling, but we don't know how this is regulated. We don't know exactly what are the mechanisms that regulate this transcription factor, either the transcription of them and even their function. So here is where we established our hypothesis, and our program is that we wanted to see in this very, very transient period, like 1 day, between hours to 1 day after the injury, is there an epigenetic mechanism that regulates the transcription of these transcription factors? And this regulation, in our view, should be dynamic, because these transcription factors they have to be able to go up and down the levels of transcription factors. So why we focused on antisense transcription? I mean, like a very brief overview is that, yes, we know that human genome is transcribed in a bidirectional manner, even though people thought for many, many years that most of the antisense transcription is like basically junk DNA in the form of antisense RNA. So we know now that this 70% of human genes are transcribed in both orientations, and obviously this is not junk, it has a significant role. So the characteristics of antisense RNA transcription is we know now that it exists as in the form of short, like microRNAs, or long antisense RNAs. A global transcriptome analysis that has been done shows that 70% of the mammalian transcriptome contains natural sense antisense pairs. The antisense RNA transcript may instruct the formation of heterochromatin repressive complexes on promoters. And with the antisense transcript, you can target multiple genes like microRNAs, which is very well known, or specific genes, which is usually the long antisense RNA way of function. It's a fast and very reversible regulation, so it's very dynamic. And the important thing is that the antisense RNAs can regulate the translation, which is how we know, how we started with, I mean, everybody's using siRNAs now, microRNAs, this kind of reagents, which regulate eventually translation of proteins, but you can also regulate transcription with antisense RNAs. The models that were published years ago, like 11 years ago, is that that antisense transcription can happen either head-to-head from like a transcription starting from 5' for this transcript, the antisense transcript, this is the sense transcript, or at tail-to-tail. So this is the 5' starting from the sense, and the antisense is here. So the transcripts actually are complementary at the 3', or overlapping within the sequence. And again, many, many years ago, like 11 years ago, people put together 4 hypotheses of how antisense transcription may work. The first hypothesis, which turned out not to be the case, is that it could be that two RNA polymerases actually collide on an sense and antisense orientation and stop transcription. Then there was papers described that maybe that antisense transcripts mask RNA splicing sites, and that changes how the transcripts are transcribed. Also, there is a possibility that interfere with RNA editing, and by having the antisense transcripts, you induce or inhibit RNA editing, which leads to nuclear retentions of the transcripts or degradation. But the model that actually prevailed, and it is studied widely now, is chromatin remodeling. And chromatin remodeling is the model where the antisense RNA either recruits epigenetic modifying complexes with histones and histone modifying enzymes, or it's part of complexes on the promoter to regulate genes. So coming to our work, is we studied both. We started with studying the role of microRNAs in nerve injury response some years ago, and we have published this data. is basically just an overview of this data, and because this is actually the segue to the latest work that we're doing, is that after nerve injury, we performed a microRNA array, and we found lots of microRNAs in this. You see it in this table, 6 hours after injury and 24 hours after injury, that are deregulated. So most interestingly, if you take these very big players like SOX2, EGR2, CJUN, NANOG, ID2, which have a major role in nerve injury response, they have multiple binding sites for these microRNAs that we found that are deregulated. So each transcript has multiple sites. So this actually made us put together and propose this hypothesis that you cannot really say that one microRNA regulates a transcript. It's a combinatorial interaction of microRNAs within transcripts in order to understand how this mechanism works. And not only it's a combinatorial interaction, something which is very difficult to interpret, but it's extremely important, is the secondary structure of the microRNAs and the RNA, the transcript, the target, plays a major role. So basically, what is the accessibility of these targets? So how much energy you need in order for the microRNAs to bind to the transcript actually dictates which microRNA is more important for the regulation of each transcript. So we did this analysis to all these major players, and we found that the element of the promoter of EGR2, which is called myelin-specific element, has this very high affinity binding sites for microRNA 709. And this created this question that is it possible that microRNA 709 binds to the MSC of the EGR2 promoter and regulates the transcription of EGR2? So we did these studies, and we showed that yes, after injury, the level of EGR2 is basically, as a protein in 48 hours, is completely gone. And as a transcript, you see it downregulated. But if you overexpress this microRNA with a microRNA mimic, what you see, and this is a nuclear unknown, that nascent transcription of EGR2 is inhibited. And then we proved that this inhibition is actually orchestrated by the microRNA 709, which brings H3 trimethylated K27 on the promoter as a complex. And this complex formation actually depends on the presence of the microRNA 709, because if you treat it with RNase H and break the RNA DNA bonds, then you don't have the formation of the complex. So basically, we showed with this work, we showed that peripheral nerve injury induces deregulation of microRNAs in sciatic nerves, and transcription factor expression is regulated by a synergistic action of microRNAs, and specifically microRNA 709 mediates the formation of epigenetic complexes on a specific, the myelin-specific element of the EGR2 promoter. So now turn to the most recent work and the recent publication, is that what is the role of long non-coding RNAs in nerve injury response? So in order to answer this question, we perform a modified 5-race protocol focusing on the promoter of EGR2, since we already had the data that there is a microRNA regulation of the EGR2 promoter. So we did this 5-race protocol and the primer working, and identified 1,000 nucleotide sequence upstream of the transcription start site of EGR2. So which we sequenced, we cloned it, and then we did all the computational analysis, searching databases to see if this is a non-coding RNA that has been reported before, and it was not. So we decided that if this is really true, it's a novel antisense RNA. Mm-hmm. So then we checked published database, RNA-seq databases, and this is actually from a paper published from the Feltri lab, is that they have done RNA-seq on mouse sciatic nerves. Mm-hmm. And we downloaded this raw data of the RNA-seq, which are available for everybody. So, and we searched for if the microRNA is expressed in this data. And we found that yes, it is, as it's supposed to be in the chromosome 10. And this is there's a 450 nucleotides, which actually constitute the mature antisense RNA complementary to the promoter of Egr2. And these are 3 different repetitions from different animals. So we're very confident, completely unbiased. These are not our data, but it's very nice to see that this exists in RNA sequencing that other people have done. So this increased our confidence that the data are real, but we wanted to see if this antisense RNA we can actually visualize it in mouse cyaric nerves. So we performed a FISH experiment, and what you see here is with the red dots are S100, which is a very widely expressed marker of Schwann cells. And the green dots are actually the antisense RNA. So this is in situ hybridization. So as you see, the expression is low, as it's expected for antisense RNAs, but it is specific on S100 expressing cells, and you can see it in the cytoplasm, you can see it in the nucleus as well. And these are the positive and negative controls. So now, the next question that we ask is there a regulation for this microRNA? Does this have a role? So we developed a strand-specific PCR. So basically, with a strand-specific PCR using different primers, you can amplify either the sense strand, which in our case is the Egr2 mRNA, or the antisense strand, which again in our case is the long antisense RNA. So we performed nerve injury, isolated the distal part of the cyaric nerve, and we performed this strand-specific qPCR. And we showed that this time course, 6, 12, and 24 hours after injury, you have, especially for 6 and 12 hours, you have a very highly significant upregulation of the expression of the antisense RNA. And at the same time interval, you have an inhibition of expression of the Egr2 mRNA. So this correlates very nicely. So actually, if you do a statistical correlation, it's like really, really highly correlated. So then we performed this serial dilution analysis from using a protocol that was published, and to identify what is the abundance of the Egr2, to identify the absolute abundance of the antisense RNA in the cyaric nerve. And we found that after injury, especially the 12 hours where you see 6 and 12 hours where you see that it's upregulated, you have close to, as an average, 500 picograms per 100 nanograms of RNA. So it's a low expression, and again, the literature agrees with that the antisense RNAs are really low in expression. And if you want to compare, it's somehow like 40 times less than the expression of the Egr2 mRNA. Okay, but now the question is, does it have a biological function? Yes, it's expressed, yes, it's upregulated, but does it have a biological function? So in order to address this question, we developed a lentivirus that expresses this antisense RNA. And when you do perform ectopic expression of this antisense RNA, you see that you inhibit the Egr2 mRNA expression. You see it here. And not only the mRNA, but also the protein expression of Egr2. So it seems just by this in Schwann cells, in purified Schwann cells, it definitely does something. And what it does, it inhibits transcription and translation of Egr2 transcription factor. So then we wanted to see if it has a role in myelination. So here you see this DRG Schwann cell co-cultures. In red, you see the myelin that is formed in a control culture infected with a lentivirus with like a non-targeting lentivirus. While when you infect these cultures with a lentivirus expressing the antisense, you have tremendous demyelination, and you see it in higher magnification here, where the myelin, you have like bits and pieces of the myelin. So it really affects myelination, and we, in order to actually improve the specificity and show the specificity of this, we developed this oligonucleotide gapmer, which is basically an oligonucleotide complementary to the antisense RNA. And if you overexpress this, it blocks the antisense RNA. So we wanted to rescue the demyelination by incubating these cultures with antisense gapmer, and we actually did. So when you culture these cells with a scrambled gapmer, which doesn't target anything, you don't rescue the demyelination. But when you culture them with a gapmer that blocks the expression of the antisense RNA, then you rescue the myelination, and you see the quantification here. So this is highly specific, and it depends on the expression of the Egr2 mRNA. So that was very encouraging. So what we did in order to increase our confidence with all this data is we formed like 5 different oligonucleotide gapmers, and we saw that if you overexpress if you give these gapmers, if you overexpress these gapmers, what happens? They block the expression of the antisense RNA, and they increase expression of the Egr2 mRNA. So they all work as it's supposed to. So we wanted to be able to deliver these gapmers in vivo in order to study nerve injury response. So we started a collaboration with Bob Langer's lab at MIT, where he developed for us, his postdoc developed for us biodegradable hydrogel, which actually it has incorporated this oligonucleotide gapmers within the hydrogel. So basically how this works, when you do an injury, when you open up and do the sciatic nerve injury, then you actually load the hydrogel, which is in liquid form, which has the gapmer inside. You load it on top of the sciatic nerve, and you keep this wound open for 2 minutes. And within 2 minutes, because the hydrogel becomes a gel, and then you close, and then this starts to degrade, and it's a biodegradable hydrogel, and it's a non-swelling hydrogel. And as it degrades, it releases the gapmer. Mm-hmm. So we have done in vitro kinetics for this, and we know that it releases gapmer for 7 days. Okay. So that was perfect, because we wanted to study this initial period of nerve injury response. So we use this oligonucleotide, the hydrogel with the gapmers to perform nerve injury. And here's an electron microscopy study where you have in 2 days, 5 days, and 7 days after injury, mice that didn't get anything, mice that were treated with a scrambled gapmer as a control, and mice that were treated with the antisense RNA gapmer. The most important, and this is quantification, myelinated, demyelinated, number of myelinated axons. But the most important thing that I wanted to focus here is that even we're talking about 1 week after, and the nerve injury that we're talking here is not a compression injury, it's a complete transection of the sciatic nerve. So basically you cut the nerve completely. So here you have 7 days, 1 week after complete cut of the sciatic nerve. And you see the control and you see the scramble, there is no myelin, there's complete degradation. But in the treated nerves, you still have axons which are completely myelinated. Of course, you have loss of myelin, obviously, this is a cut nerve. But the fact that you can see axons with complete myelination still, a week after complete resection, it's just astonishing. Just by inhibiting an antisense RNA, you can do that. Mm-hmm. So this is really, really important. So, of course, then this creates a lot of questions. How does this work? And the first thing that we wanted to prove was that this really affects transcription of Egr2, because we haven't proved it. And in order to do this, we did this nuclear unknown experiment where we showed that the expression of the antisense RNA inhibits nascent transcription of Egr2. So it really targets to Egr2 transcription. And also, in order to connect with this data, to see that really we have the sciatic nerve retains the major structural myelin proteins, we did a time course of Western blot after the addition of the antisense gapmers or the scrambled gapmers, and you see in 7 days that both myelin protein 0 and PMP22, the structural proteins of peripheral myelin, are really upregulated. They are still, actually, in order to say it correctly, they are still not inhibited when you treat with the gapmer and you block the antisense RNA. So this is real, and this inhibition of the antisense can have the ability to actually delay the myelination after injury. So go to the mechanism and understand how this works. So there are papers that have been published the recent years that ERK1/2 signal entity is known also that after injury, the neuregulin axis is disturbed, of course, and this affects ERK1/2 signaling within the Schwann cells. So the ERK1/2 regulates expression of EGR2. And in order to, other people, John Zvaren's lab has shown this, Patricia Kazakia's paper, they have shown this, that when you inhibit ERK1/2 and you inhibit it with the UO126 inhibitor, what happens, you inhibit EGR2 expression. So there's direct link between ERK1/2 signaling and EGR2 expression in Schwann cells. So what we wanted to see, if we treat this with the Schwann cells with UO126 and block ERK1/2, what is the effect on the EGR2 antisense RNA? And what we found is that when you block ERK1/2 signaling, yes, this is a qPCR data. The EGR2 mRNA is inhibited, as it was shown by others, and it's significantly inhibited. But the expression of the antisense RNA is actually activated, and it's significantly activated. So it seems that ERK1/2 signaling has, in a normal Schwann cell, has a role to keep the antisense RNA in a repressed state. When you inhibit ERK1/2 signaling, then you have de-repression of the expression of the antisense RNA and inhibition of the EGR2 mRNA expression. So how does this work? So in order to understand this, we thought, again, based on the work of John Zvaren and Patricia Kazakias, the Nature Neuroscience paper that they had, they connected the expression of EGR2 ERK1/2 signaling to YY1. So we thought that we will study the same with the role of YY1 on this regulation, based also on another aspect. And this aspect is that if you really look closely at the sequence of the antisense RNA upstream of the transcription start site, so you see that very close to the start of the antisense RNA sequence, there is this motif, which is a YY1 binding motif. And the interesting thing for this motif, it's actually YY1 binding in both directions, both the sense and the antisense orientation. And this little part of the sequence, which is close to 100 nucleotides, has all the elements of a bidirectional promoter. So we thought that maybe this part of the antisense RNA has a role, which is part of the EGR2 promoter, has actually a role of a bidirectional promoter for the antisense RNA as well. So we focused on what, and the only transcription factor binding site on this part of the antisense RNA is this YY1, bidirectional. So we focused on this site, which we called S1 site. And we also focused on a different site, not focused, we used as a positive control, let's put it this way, an S2 site, which is further upstream. And it has been shown in the literature that it's a real bona fide YY1 binding site. And this is just to confirm that we're doing the right thing. So we perform a DNA chip for YY1 with and without the addition of the U126 to inhibit ERK1/2. And this is a YY1 chip on the EGR2 promoter, DNA chip. So we saw that, yes, when you inhibit ERK1/2 signaling, you have partial inhibition of YY1 binding to the S2 region, to the S2 motif, which is our positive control, which is shows that the technique is working. And there is no binding either with the control or the ERK1/2 in the S1 on the DNA, which is fine. It actually agrees with the literature, this result. But when you do an RNA-IP to see if YY1 interacts with the antisense RNA, okay, what's very interesting is that in control Schwann cells, it has almost no interaction with the RNA. But when you inhibit ERK1/2, YY1 significantly increases binding to the S1 site and the S2 site, but mostly to the S1 site. So it seems that inhibition of ERK1/2 signaling induces binding of YY1 to the antisense RNA on this specific YY1 binding motif. So, which is great. So what is the role and how does this happen? So why do you need to inhibit ERK1/2 signaling? What's going on? So if you look at YY1 in general, the regulation of YY1 depends on post-translational modifications. And people have shown that YY1 can be phosphorylated, can be acetylated, can be ubiquitinated. So we focused on phosphorylation since it's a result of kinases, or could be a result of intracellular kinase. So what we did is we constructed several mutants on several serines that YY1 has. So basically, we mutated from serine to alanine, so they cannot be phosphorylated. The 118 serine, the 184, the 247, and then we did double mutant, the 118 and 184. And we performed YY1 RNA IPs, both with the wild type, non-mutated YY1, and each of these mutants. So what we found is that when you actually delete serine 118, you inhibit binding of YY1 to the antisense RNA. But the most interesting part is when you delete the serine 184, so it cannot be phosphorylated there, then you have a huge increase on the binding of YY1 to the antisense RNA. And the combination of this double mutant where you delete both 118 and 184, it's still upregulated compared to the control, which to us means that the 184 is the dominant regulatory site, which potentially, if it's dephosphorylated, it actually increases the expression of YY1. The binding of YY1 to the antisense RNA. So these data actually are summarized here, where we proposed this YY1 phosphoswitch as the regulatory step for the control of the antisense RNA expression. So basically, what we think is happening is that in a non-injured Schwann cell, where everything works fine, and you have the neuregulin binding to RB2, RB3 receptors, activating ERK2, then you have phosphorylation of YY1, which represses the antisense RNA and activates the expression of Egr2 mRNA. But when injury happens, this ERK2, the very initial step is that the ERK2, you have a very transient block of ERK2 signaling, which probably the hypothesis that activates a protein phosphatase that actually dephosphorylates YY1 specifically on serine 184, and the dephosphorylated YY1 binds the antisense RNA with high affinity, activates the expression of antisense RNA, and at the same time inhibits the expression of Egr2 mRNA. So this is the model that we proposed. So to summarize all the data, both for the microRNA and the long antisense RNA, we presented data to actually establish an antisense RNA code, if you will, during PNS injury response. So what we think is happening is during differentiation, you have expression of genes like c-Jun, SOX2, GDNF, and Sonic-Hedgehog, which are actually should be inhibited. And the mRNAs involved in this inhibition from papers that have been published and from our own work are listed here, and the Egr2 antisense RNA is also involved. Then during injury, you have a concerted action of both microRNAs and the antisense RNA to regulate the transition from a differentiated to de-differentiated state. And this is a dynamic regulation. The major players is the Egr2 gene and the c-Jun. And we have preliminary data to show regulation of c-Jun as well for antisense RNA. And we know that the microRNAs that are involved, we proved it at 709, 140, from the work of Jeffrey Milbrandt's lab as well. And now we also know that the Egr2 antisense RNA is involved in this dynamic regulation. So currently what we're studying, and we have some very interesting data, is that we're trying to do a phosphoproteomic analysis of YY1 to identify other possible sites that are phosphorylated or dephosphorylated during nerve injury. And how do these phosphosites regulate binding to the antisense RNA, where we're trying to do phospho-YY1 RNA IPs, which first of all, we have to develop good antibodies for that. And this is what we're trying to do. So we are already started a genome-wide H3K27 and H3K4 trimethylated chip sequencing after inhibition of the Egr2 antisense RNA to see if you have a transition between repressive and activated complexes on specific promoters, that these complexes are actually orchestrated by the antisense RNA. We are also started and we are already developed this CHIRP method, which basically what you form, tandem oligos that actually cover the whole area of the antisense RNA, and these are biotinylated, and use them as a bait to bring the antisense RNA, to precipitate the antisense RNA together with the proteins or DNA that interacts with that. So we're actually doing this in order to identify globally what are the interactors, both as a protein and as a DNA, that this antisense RNA interacts. And I want to say that we're doing this because we think that there is the antisense RNA acts obviously in cis to regulate Egr2, but there is a very high, it is very likely that it can work in trans as well, and it has been proved for other antisense RNA, like for X chromosome inactivation, that you can have antisense RNA that work across chromosomes. And we want to see if this antisense RNA that we identified can do the same. And this will actually lead to intervention studies where we can actually inhibit specific chromatin modified enzymes to see how this regulates nerve response after injury. And we also want to do some behavioral studies to see if we modulate the expression of the antisense RNA and we modulate these chromatin modifying enzymes. Do we change pain responses? Do we change myelination? Do we change the degree of the myelination or degree of regeneration? This will be very interesting to actually prove if our studies for the antisense RNA could have a translational flavor. And I will stop here. I want to acknowledge the people in my lab, mostly my postdoc Margot, who is the first author of the last paper that we published, who has done most of the work for the long antisense RNA, but the other people in the lab as well, our collaborators and the funding agencies as well.
So if you can unshare the presentation, sure. I have a couple of questions.
Yes.
These are like real basic early things. One is you showed all these phosphorylation and dephosphorylation of YYY site and all. Do you know what is the signal goes there? Like how does these protease know what to do? And what is the signal as soon as the denervation happens, that goes there and hey, you need to phosphorylate or dephosphorylate? Do you have that early signal which is sent out from the injury site?
Yes, so we think that the early signal, so we have some preliminary evidence for that. So we think that the early signal is not actually, a signal that in a terms of how you think that is a receptor-ligand interaction. Actually, it's the opposite. It's the fact that, so it sends a signal through what we have so far, through the neuroregulin RB2, RB3 axis, that this axis is disturbed. Okay, because the action is probably the first element in the sciatic nerve to signal that something wrong is happening.
Yep.
So the disruption of neuroregulin RB3 interaction is the first thing, the first signaling event that we have noticed that is happening in Schwann cell. Mm-hmm. So very early, to give you an example, very early, when I'm saying very early, is 4 hours after injury in vivo. So we did the injury, and 4 hours we take the nerves. You have a deregulation of neuroregulin ERK1/2 signaling already. And you already have activation of the antisense RNA. And you already have activation of specific phosphatases in the Schwann cells. So that's not a complete answer to your question, because there could be other receptors, of course, that can be deregulated after injury, but we haven't done, and we don't know this early what other receptor or ligand interaction is disturbed.
Okay. And other thing is that you had mentioned like there is a complete, when you do the experiment, it is the complete denervation, right? Like you just chop off the nerves.
Yes.
And like you showed with the hydrogel that there is still like connection built up when you do the add those hydrogel with the oligos or antisense or whatever you have used. So is this can be like you see similar thing in the spinal cord injury, like there is a breakdown of spinal cord, like so can you like see the similar mechanism? Like it is just like hypothetical question.
It's very difficult to say for the spinal cord. So we obviously at some point during these years doing this work, we wanted to see if Egr2 is expressed in oligodendrocytes. Okay, but it's not. So maybe the same kind of mechanism works, but it's not the same antisense RNA.
Yeah.
So now what is interesting is that there is work that is being done that using scaffolds or hydrogels bridges between the injured sites in the spinal cord, and it has some encouraging results there, that it helps regeneration or remyelination or establish some new connections. But I think that this is a very tough question to answer. And the antisense RNA, the role of antisense RNAs in oligodendrocyte myelination or remyelination, yes, it's something that needs to be done.
Okay. Yeah, that's it. I think it's an excellent talk, and I learned a lot about it, about your work, and it will be on our website as soon as we are done with that. Like I'll send you a link. And
Okay.
Is it okay if I share your email address to all the Facebook, LinkedIn, like if at all they are interested in your work, they can contact you?
Yeah, the Brown EDU email, yes.
Yeah. Okay. So I'll do that. So if at all anybody is interested, collaborate or looking for a position or anything, they can get back to you, and you can go from there. Again, thank you very much. I really appreciate you doing this, and hope to see more of your work in the near future.
Good. Thank you so much for the invitation. Thank you.
Thank you.